So we left off at the end of video 1 looking at a brief summary of the 2 stages of division that we see in meiosis. We saw that meiosis 1 splits our homologous pairs, so the 2 cells at the end are going to be haploid. They don't have a mom and a dad. They don't have those 2 copies of a chromosome. They just have one. But my chromosomes at the end of meiosis 1 are still duplicated. They're still those X shapes. So that's why we have a second round of division where we split your sister chromatid. So you separate those X shapes and you're going to put one unduplicated chromosome into each of the 4 cells you make at the end. So I end up with 4 haploid cells at the end of meiosis. What we're going to do now that we've just super briefly gone over the big picture, is we're going to focus in here on metaphase 1. So metaphase 1 was where we had my homologous chromosomes. So my duplicated mom version of a chromosome and my duplicated dad version of a chromosome. And they're sitting next to each other when they line up during metaphase 1. And 2 interesting things occur in this phase that help us to understand about some of the variation that this process introduces into eukaryotic sexually reproducing organisms. The first thing that we notice here is we have in this lining up of our homologous pairs, so my mom and dad copy of the same chromosome, is you have what's known as the law of independent assortment in action. What this means is that when my mom and my dad pair line up during metaphase 1, which side they're on, so whether my mom copies on the left side or it's on the right side, is 100% random. Every single time they line up like this. What that means is if you look at the diagrams up here, over on my left-hand side, they're showing dad DNA in blue and mom DNA in red. So they're saying it's possible that I get all of my dad's chromosomes lining up on one side, mom lining up on the other. So dad DNA goes this way, mom DNA goes the other way. It is equally possible that my first pair, my dad chromosome is on this side, mom chromosome is on this side, and in my second pair, mom's on this side, dad's on that side, which means I am equally likely, as I am to get this setup, to get cells at the end that have one copy of dad's chromosome number 1 and one copy of mom's chromosome number 2 here, and one copy of mom's chromosome 1 here, one copy of dad's chromosome number 2 here. Equal possibility. Now we're only looking at 4 chromosomes here. Remember that with humans, you have 46 chromosomes, which means that for every single one of those pairs, every single one of those 23 pairs you have, it's equally likely you're going to get the mom or dad version every single time these cells line up to do meiosis, which creates a tremendous amount of variation between the types of gametes, the types of sperm or egg cells, and what combination of genetics is going to end up inside those cells we make. Because every single time sperm is formed, every single time an egg is formed, you have a random lining up of each of your homologous pairs. So that's why no 2 sperm, no 2 eggs are going to be genetically exactly identical to each other. The second thing that happens during metaphase 1 is something called crossing over. So here's the cool thing about when you have your dad copy and your mom copy, your homologous chromosomes, sitting next to each other in metaphase. Remember that the reason that mom and dad are homologous is you got one exact copy of info from one parent and one from the other. But they're homologs, which means same location, meaning they've got the same genes in the same places. So if you look at this diagram over here on the left, they're showing dad is dominant for A, dad is dominant for gene C, and dad is dominant for gene C. So A, B, and C, dad is all dominant for. Over on mom's chromosome, she again, because they're homologs, she has the same genes, so a gene A, a gene B, a gene C, but she's recessive for each. What can happen during this phase is that when the chromosomes sit next to each other, sometimes they get a little tangled up in each other, and they can actually swap pieces. The exchange of pieces between homologous pairs like this is called crossing over. Notice what happens here if you look over to the right-hand side of the diagram. I didn't lose any genes. So if you look, I still have an A, B, and a C, and an A, B, and a C. But I get a new combination of genes. So on my dad chromosome now, I've got dominant A, dominant B, and recessive C. On my mom chromosome, I've got recessive A, recessive B, and dominant C. So crossing over increases the variation. This is how it is that you can end up with information from 2 different parents for a trait that are on the same chromosome, but you got some info from mom and some info from dad. So it actually makes entire chromosomes that are a blend. And this is a completely random process. It does not always occur. We cannot predict very accurately where it's going to occur. Some spaces are more likely to do it than others. Genes closer to each other are more likely to have it occur than the genes that are further apart. But it is random. Quick review. So first question, what is the end product of meiosis? Good. So unlike mitosis, which makes 2 diploid cells, meiosis makes 4 haploid cells. Which type of organisms perform meiosis? Good. So that is only sexually reproducing eukaryotes. Asexually reproducing eukaryotes, they'll just use mitosis to reproduce. Meiosis, where you create sperm and egg, you only need to create sperm or egg if you are going to perform sexual reproduction. Remember, bacteria only do binary fission, so they are never doing mitosis or meiosis. So again, quick recap, check to make sure you've got all of this info in your notes. What we were looking at here is just a quick cursory overview of how sperm and egg get created. We'll do more of this in class together, but we wanted to identify sources of genetic variation. So you should know law of independent assortment, what it is, how you find it, and then also crossing over, how these are sources of variation, what they are, and how they occur. Please see me if you have any questions.